Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 6 for January 2023. Let's begin with the first question. Question 1. The photograph shows Lambda minor, a species of aquatic plant. So these are the plants they're talking about. This is one plant and it has 3 leaflets. They say these plants live in ponds, lakes, and slow-flowing rivers in Asia. These plants absorb nitrate ions and grow rapidly. Each plant develops 3 leaflets and then divides to produce a daughter plant. In optimum conditions, the plants divide every 2 days. So when you look at this information, there is the idea that they absorb nitrates. That's why I circled some of the things or underline them, because they're going to be important in our next question. So the key things are they absorb nitrates, they grow rapidly, they have 3 leaflets, and then the division occurs every 2 days. So moving on. Here they say, describe an experiment to investigate the effect of nitrate ion concentration on the rate of growth of these plants. In every biology question where they ask you to describe an experiment, it is very important that you state the dependent variable and then how the independent variable is going to be varied, how the experiment is going to be repeated, how you are going to measure the dependent variable, in what amount of time is the experiment going to be carried out, and then which variables you're going to keep constant and how you're going to maintain them constant. So I said the dependent variable is the number of plants produced, or you could say the mass of the plant. In this case, if you're using the mass of the plant, then in the end you're going to weigh the plant before and then weigh it after. Or if you're counting the number of plants, because division occurs every 2 weeks, you'll see how many plants have divided, and therefore you'll know the effect of nitrate ion concentration. Then you can set up 5 different concentrations of solution of nitrate ions in beakers. This is the independent variable and how it's going to be varied. The plants used should be of the same species, they should be the same age, and they should be the same number per experiment. Then allow the plants to grow for the same period of time. It could be 2 weeks or 4 weeks. Then count the number of plants produced per time. This is how I'm going to measure the dependent variable. So this could be per 2 days. And then control the temperature using a thermostatically controlled water bath and control pH using a buffer. Then repeat the experiment and calculate the mean, and then calculate the rate of growth by dividing the number of plants produced by the time in which those plants were produced. So if you had written something close to this, you will get all the 6 marks that were awarded. Moving on. Here they say, explain the roles of nitrate ions in cell division to produce daughter plants. From this part, you can see we need nitrate ions and how they lead to cell division. Nitrate ions are used in amino acid synthesis, in enzyme synthesis, and because enzymes control most of the other processes that occur within the plant, then it means they're going to be very vital. So I said they provide nitrogen for formation of amino acids, enzymes, chlorophyll, and so on. And they are useful in formation of nucleotides for DNA as well as RNA. So this brings us to the end of question 1. Let's continue to question 2. Question 2, the photograph shows bacterial cells of E. coli as seen using an electron microscope. We can see these are the cells. They say the growth of bacteria in a liquid culture can be investigated. The number of bacterial cells in the culture can be measured using the dilution plating technique. The first part says, identify one risk that may be encountered when carrying out this technique and how this risk could be reduced. The major risk is E. coli could be pathogenic, so I said E. coli are pathogens. And of course, these are 2 marks, so 1 mark is for this and the next mark is for this. How this risk could be reduced? Of course, use aseptic conditions, like minimum lifting of the Petri dish lid during the experiment, and use gloves to avoid direct contact. Also, culture the bacteria below the body temperature, because if you culture above or at the body temperature, then you're giving a chance for the pathogenic bacteria to also be cultured. So moving on. Here they say, the growth rate constant K for a bacterial population can be found using the equation K is equal to log 10, NT minus log 10 NO divided by 0.301 times the time. So NT is the final number of bacterial cells, NO is the initial number of bacterial cells, NT is the growth period in hours. So here they say the initial number of bacterial cells was 900, so this is going to be NO. And after 2 hours, so 2 is the time, there will be 14000 bacterial cells in the culture. So this is our NT. They say calculate the growth rate constant, K, and give your answer to 3 significant figures. So I substituted, this is quite easy. I wrote log to base 10 of the NT, which is that, minus log to base 10 of 900, divided by 0.301 times the time, which is 2. And remember the time had to be kept in hours. So the rest is just fed into your calculator. And then in the end, I rounded off to 3 significant figures, which is 1.98 as my answer. Moving on. Part C, state 2 abiotic variables that could affect the growth rate of these bacteria. Of course, the first variable is temperature, and the second variable I chose pH, or you could choose oxygen concentration because aerobic respiration could be occurring. And the next part says, choose one of the variables you have identified in C1 and describe how this variable could be controlled and the effect it could have on the results if it's not controlled. I chose the variable which is temperature, and how could this variable be controlled? Using an incubator during the culture. And then to describe the effect this variable could have on the results if it's not controlled, the results will not be valid because temperature is very vital. If it goes so high, then the enzymes are going to be denatured, and if it's too low, then the rate of reaction is also going to be slow. So this brings us to the end of question 2. Let's continue to question 3. Question 3, the photograph shows a marine worm which is extending its body from a burrow in the sand. So this is the worm. So down here they say the animal is found in the Sea of Japan. This animal feeds on organic particles in mud and sand habitats. They say a scientist observed that these animals respond to a sudden increase in light intensity by withdrawing into their burrows. The animals then extend their bodies to continue feeding. The scientists selected 11 animals living in mud and 11 animals living in the sand. So here, it's like we're carrying out a comparison between the animals in the mud and the animals in the sand. So when you're studying the null hypothesis, it could be there is no significant difference because you're comparing two things. So they say each animal was exposed to one burst of bright light. The time in seconds before beginning to extend their bodies from their burrows was recorded. So we can see these are the results for those in the mud habitat and the results for those in the sand habitat. Moving on. So here they say state a suitable null hypothesis for this investigation. Because we were comparing two things, it means there is no significant difference. I said there is no significant difference between the mean time before body extension among organisms in the mud and those in the sand habitat. The next part says draw a suitable table to display the data and your calculated means for the time before extension in mud and sand habitats. The key thing here is they want you to show a table that has units as well as labels, and the means have to be calculated. So I wrote time taken before extension in seconds. For the mud habitat, I wrote the results as raw data, and then for the sand habitat, they're also as raw data. And then down here, I calculated the means. To calculate the mean, we have to add up everything here and then divide by the numbers of how many they are in that group. We did the same thing for this side, and my mean here was 11.5, while this was 10.3. So the table is as you can see here. Moving on. In part B, draw a suitable graph to display your calculated means and the variability of the data for these two groups of animals. Now again, because here we're comparing two groups of animals, and like I already said, the major thing here is not a correlation, but we're trying to compare if there is significant difference. The graph we're going to draw is going to be a bar graph. One part is for the mud habitat, and the other part is for sand habitat. And also in the question here, they've told us about variability. It means we have to include error bars. So to include error bars, we have to go back to our data here, look for the highest value here and the lowest value in that group. And then here we look for the highest and the lowest value, and we use them in putting the error bars on the graph we have drawn. So here you can see I drew the mean for this part. Let me try to show you the whole graph. So on the vertical axis, I have time taken before the extension, and on the horizontal axis, I have habitat. And then for the mud habitat, this was the mean value around here. and this was the lowest value, and that was the highest value. I connected them. And for the sand habitat, the mean value was that, and then the lower value was that, and the highest value was that, and I connected them. The key thing here is you have to show the X and Y axis, and they have to be labeled with units, and then you have to get marks for drawing a bar graph as well as putting the error bars as required. Moving on. In part C, the student analyzed the data with a t-test using the formula. So this is the formula they used. They want you to calculate the value of t. So I used the result from my table, of course, because they say this is the mean. From the table, I had that mean as well as that mean. So I subtracted the higher mean minus the lower mean. And then down here, I got the first value here and I divided by the number of samples that were collected. And the second value here divided by the number of samples that were collected. And fitting everything into the calculator, I got 1.374. And rounding off to three significant figures, I got 1.37 as my answer. So that's what I wrote here. Moving on. They say the table shows the critical values of t for different degrees of freedom. They say the number of degrees of freedom can be calculated by saying NA minus 1. This is going to be 11 minus 1 plus 11 minus 1. So this is going to be 10 plus 10, which gives us 20. So it means we're going to get our result, or the critical result, at p is equal to 0.05, corresponding to 20 as the degree of freedom. And we can see this is the value that corresponds to that. So our critical value is 2.09 from the table of critical results. Let's move on. Then here they say, explain the conclusion that can be drawn from this investigation. They want you to use your graph, your calculated t-value, and the table of critical values to support your answer. So you have to state the critical value. So I say the critical value is 2.09, and the calculated value is 1.37. So this calculated value is lower than the critical value, so we have to accept the null hypothesis. And that means there is no significant difference between the extension time of organisms in the sand habitat and in the mud habitat. So I also put something talking about the error bars. The error bars do overlap, so the result is not valid. Down here, they say this animal has light-sensitive pigments called opsins. Suggest how opsins allow this animal to respond in this investigation. I said in the presence of light, the cis-retinol in opsin is converted into trans-retinol. Then this process leads to a cascade of reaction that occurs, producing a generator potential, and this is converted into an action potential. From here, impulses are sent to the central nervous system, and a response is sent back to the muscle in a reflex action to cause muscle contraction as well as movement. So marks were awarded for writing about a reflex action as well as talking about the process of conversion of the cis-retinol into trans-retinol, and then the generator potential as well as action potential generation. So this brings us to the end of question 3. Let's continue to question 4. Question 4. The photograph shows a flightless grasshopper. They call it the Brachyspis robustus. This is the grasshopper they're talking about. They say this endangered species is only found in one small part of New Zealand. These grasshoppers are approximately 4 centimeters long. The grasshoppers are most active between November and March. The grasshoppers are camouflaged, so they can only be seen when they move. A student decided to compare the population of grasshoppers living on an unused gravel road and a natural gravel area. The student formed the following hypothesis. The population of B. robustus in the natural gravel area will be larger than the population on an unused gravel area. So, here they say, plan an investigation to find evidence to support or reject this hypothesis. And then they want you to describe preliminary practical work that you might undertake to find a suitable method for observing these grasshoppers to provide quantitative results. Before I answer this, I want to take you back here. These grasshoppers are camouflaged, and we have to find ways to make these grasshoppers to be able to move in order for them to be seen. Again, they talked about the months, November and March, so we have to find suitable months or a day with the highest number of grasshoppers to perform the study. And also we have to find a suitable method to identify the species of grasshoppers. We have to find a suitable sampling method. Are we going to use quadrats? Are we going to use nets? We have to do that. And then find a way to stimulate the grasshoppers to move, because if they're camouflaged, they won't be seen. So make them move so that you can be able to see them. Moving on. Now this is an 8-mark question. Devise a detailed method, including how you would control and monitor important variables. And again, this question here is unique. It's not as the most common questions, whereby you have to vary the independent variable and so on. So here we have to use quadrats, or you could use a net in order to count the number of grasshoppers per specific area. And you have to do it randomly. So I said the dependent variable is the number of grasshoppers per unit area. We have to select suitable areas. It could be a natural gravel area and an unused gravel road. Their dimensions have to be known. You could use quadrats that have nets. In this way, as the quadrat is thrown, the grasshoppers that are held within that area are not going to be able to escape, so you can be able to count them. So use quadrats with a net to count the number of grasshoppers per specific area within a specific period of time. And then monitor temperature using a thermometer, light intensity with a light meter, and pH with a pH meter. These are the variables that we have to make sure they are controlled so that they are not changing so drastically in the different areas we are trying to investigate. Then ensure that the gradient or slope of the chosen area is the same. You have to repeat experiments in other sample areas under similar abiotic conditions and calculate the mean. If you're using a gravel area, observe the gravel area as well as the road. So you have to repeat for each and then calculate the mean number of grasshoppers. And repeat the investigation at different times of the year, or it could be different seasons, and then calculate the mean. And finally, calculate the mean number of grasshoppers per area by dividing the mean number of grasshoppers by the specific area. Moving on. Here they say, describe how your results should be recorded, presented, and analyzed in order to draw conclusions from your investigation. So here I chose not to draw anything because maybe sometimes it's misleading to some students. You are not required to draw a table or a graph, provided you can write it in detailed ways. So I just wrote. I said present the raw data in a table with headings and units. Then repeat the experiments and calculate the means and include these means in a specific column. Then draw a bar graph with labels and units. And then use a specific statistical test like the t-test to test for a difference. Moving on. Suggest 3 limitations of your proposed method. I said it's difficult to see the grasshoppers if they camouflage. It is difficult to know which grasshopper has already been counted or not. It is difficult to control the temperature, the pH, and other conditions in nature because different areas could have different changes in those conditions. And the populations of grasshoppers could rapidly change in an area since they are first moving. This could be seen like in the morning, they could be different, and in the afternoon different, and in the evening they could be different. So you have to make sure you are monitoring at the exact time for all areas to have results that are valid. So this brings us to the end of question 4, as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.